Hello, everyone. My name is James Polly, and welcome to the quarterly CERSI series, the Center of Excellence in Regulatory Science and Innovation. My name is Jim Polly. I'm a faculty member here at the University of Maryland, and I'm really pleased to be able to speak with you on this CERSI series on prediction of oral drug absorption and biopharmaceutic risk using a dissolution hollow fiber membrane D-HFM system. And this is an example of a project involving the Maryland CERSI with FDA. And let me just say thanks to Dana and Anna, who are at the University of Maryland. And Dana's doing IT, and Anna's going to be monitoring the chat. So feel free to enter questions into the chat, and we'll have plenty of opportunity to take questions. It's a little bit late here on the East Coast. It's very late in Europe, and maybe even very middle of the evening for those in the east of the world. But thanks for those who might be up and about at this time from that part of the world. And so Asmita is a former graduate student. She and I worked with Paul So at the FDA on this project that recently wrapped up earlier this year. And really just talking about two publications from this work. They're both cited here. And be glad to get you, if you email the CERSI website, the MCERSI website, glad to send these slides on to you. These are both in the Journal of Pharmaceutical Sciences, another way to get these articles. And the two topics involved in the first one, characterizing a dissolution hollow fiber membrane system, which we call D-HFM, and compare the resulting in vitro drug permeation constants denoted K prime P. So sort of a rate constant we get from in vitro studies involving a dissolution permeation system to in vivo clinical permeation constants that we can get from clinical PK studies denoted KP for drugs that span various characteristics, so-called BCS, of biopharmaceutics classification system classes. And the next one, this second part of the talk this afternoon, predicting the in vivo human performance profile and absorption profile and biopharmaceutic performance of 5 products using this in vitro system, for which we had PK data. So the scope was to look at this reasonably new system, although it's been in the marketplace for quite some time, as a dialysis membrane, and subject it to do some characterization about it, some basic characterization, understand it, and then subject it to some products that have already been studied pharmacokinetically. And just acknowledgement and disclaimer, so this has been kindly supported through the FDA, through MCERSI. Here's a website to learn more about the CERSI network. These are just my personal views, and I'm also a member of the Scientific Advisory Board of Simulations Plus, which involves software. So, the space for this sort of work, I think many of you online will readily recognize sort of the chemistry manufacturing control area, also denoted the quality or sometimes the non-clinical area. And it concerns, attributes of drug products as well as manufacturing. And CMC is an integral part of pharmaceutical product applications to the FDA, whether they're new drugs or abbreviated drug applications. And that CMC kind of lives with the entire product life cycle, including, of course, post-market, where there's lots of changes. So we often think about the drug product, understanding the drug product, which is kind of what underpins this entire presentation, trying to understand the drug product, at least in terms of absorption aspects, which is impacted by manufacturing process, elements of quality control and release testing, and there's specifications and stability that involve drug product CMC issues. And of course, there's also the manufacturing facility, the design, qualification, operations, and maintenance. So just some key points in the CMC areas that, product understanding is really important, which motivates this lecture. And that sort of another way of thinking is that tablets and capsules don't come from prescription pads alone. There's huge endeavor behind making good quality products available. And here's an illustration of the CMC life cycle. So time progresses and starting at the bottom and goes up, where early on in the development program, there's prototypical formulations that are subjected to pharmacokinetic testing, at some point human testing. if things are successful. And then there are clinical trial formulations, which could very well differ from development formulations that are assessed for safety and efficacy. And then you can even have tweaks, formulation tweaks for further formulation development that are actually the subject of new drug applications, maybe even after phase 3 testing. And then if approved, then you can have scale up and post-approval type of formulation changes, leading to supplements. And then at some point, there'll be multiple manufacturers, generics, the ANDA process. So there's many of you who work in CMC know that change is sort of the constant, and there's a frequent need to assess ongoing product quality and sameness over years. So having good product understanding kind of can really help do that. Focusing on the subject of this lecture on oral drug absorption from tablets and capsules. The most simplistic model we can think of is that we have a tablet which undergoes dissolution, resulting in drug in the gastrointestinal tract, which then is a source for drug intestinal permeation, such that we get drug that's absorbed. But there's certainly many poorly soluble drugs, and including drugs where the solubility is so low that it's maybe at some level kind of surprising how well they're absorbed. And the thinking that there's really a dynamic interplay between this dissolution step and the intestinal permeation step, where we might get a saturated solution in the gastrointestinal tract, but because of particularly drugs with favorable permeability, that then allows for more drug to actually dissolve. So there's actually sometimes quite a dependence on dissolution, on permeation, even though when we draw things out, linearly at least, that dissolution comes before permeation. But the two can be very much related. And because of that, and I'll show a slide very shortly here, there's been sort of a growth of at least testing of new systems, these new dissolution permeation systems, trying to get at this and study this, understand products a little bit better. In fact, this was definitely a topic of some discussion at a May 2023 MCERC workshop on drug dissolution, oral drug absorption, a workshop report, which was just recently accepted in the APS journal. So I think any day now that should be available there. And so one of the coming out of that discussion, not just relating to dissolution and permeation, although that was one component, is just the observation that there's been an identified an obstacle to non-compendial methods, and we often have dissolution alone as a compendial method. But looking at other things than compendial dissolution testing, an obstacle is just the uncertainty of global regulatory acceptance of such methods. And at that same meeting in May, this is an adapted slide, largely presented by Annette Bauer-Brandl. And what's going on here are all these different dissolution permeation setups. And if you ask for slides, we'll be glad to give you these. And the one I just want to highlight here is the one I'll be talking about, originally introduced in terms of drug absorption analysis by Hate a couple of years ago. But there are many, many others in the last couple of years. Okay, and then this dissolution hollow fiber membrane system, what does it consist of? I'd say there's really two features. One is dissolution testing, like I think many of you are familiar with, and then sort of hooking that up with this hollow fiber system, which has been around for quite some time. It's often used for protein purification. And this is an illustration, but there's actually 36, for the one that we purchased, there's 36 of these fibers in parallel. The blue is sort of the, I'd say from a GI standpoint, since we're trying to mimic that, the blue represents the luminal GI lumen, where here's luminal fluid as represented by a dissolution vessel. It's then pumped into the middle of a fiber, including 35 other parallel fibers. And it's recirculated back in case it's not absorbed, the setup that we've performed. And then on the outside of those fibers, sort of representing the absorption side, we can have a receiver buffer, denoted here in pink, and that's being pumped, and that way we can have a sink conditions in terms of the blood side of things. So here, this is just an illustration of this hollow fiber system. as integrated with a dissolution apparatus, basically. Okay, and just to kind of summarize this initial, well, paper, I guess, since we're talking about two papers from this MCERC project, we wanted to characterize this dissolution hollow fiber membrane system. As in the last slide, we talked about flow a little bit. We applied a couple different model flow systems, and the mixing tank model, the so-called mixing tank model, which is a rather facile system. Was perfectly fine for this. Here's the representation of that, just kind of just a simple one compartment mixing tank, basically, for this D-HFM with recirculation. One major motivation of this, and we'll talk a little bit about this, is that it had a favorable area to volume ratio. That is, the area of all those fibers is actually relatively high, normalized for volume. And I'll talk quite a bit more about that. The whole background of this is we wanted something where we can subject tablets and capsules to biopharmaceutic risk assessment. In vivo, we have a really high area to volume ratio, and what's available even right now is less than what we're looking for. But this is at least favorable, and as are some of the others that are in a previous slide. In terms of the immediate goal, was to compare the in vitro rate constant coefficient from the system to an in vivo rate coefficient from clinical studies for 4 drugs of various properties. And I would say they were close. We can take a little bit more closer look at that. I'm sure there's room for improvement, no doubt. So just, I would say we were optimistic about HFM module having potential to be something useful. That's kind of where we are today, studying these relatively simple dosage forms. Of course, the goal in the future, I think, is to study more complex dosage forms. A little bit more about this area to volume ratio. Again, if we want to, again, we're kind of motivated because how permeation can really allow for dissolution, and we suspect that happens in vivo. So if we want to mimic that in vitro, we need a permeation component, which is definitely dependent on area to volume type ratios. So for this HFM system, it has an area of 115 centimeters, which is quite big for an in vitro system. And then if we use 100 mLs, then that ratio is 1.15. In terms of what are some estimates in vivo, some estimates have been on the order, actually around this, maybe a little bit higher. There's been an estimate from our lab around 11 per centimeter, and a couple other labs around 2.2 and 1.9. And if you want to start thinking about this sort of thing, what do we mean by some of these first order rate coefficients? You can get a first order rate coefficient by considering the permeability of the drug across the membrane, and then multiplied by this area to volume ratio. So for example, area to volume ratio in the HFM system, if you have 100 mLs as your donor volume, it's going to be 1.15 per centimeter. And another concept that you can start thinking about is, okay, well, what should our target be? Let's say if we happen to have a system with a certain permeability in vivo, and the apparent permeability in vitro is a certain number, what should that be? Okay, so in terms of this hollow fiber system, first thing we wanted to do was actually subject it to solutions. And we just wanted to start doing some characterization, some predictive characterization with a big emphasis on predictive. So there's a certain, this membrane, this hollow fiber system is made of a certain plastic. So we're able to buy plastic that's identical to what the system is made of. We did some so-called flat sheet permeability studies, which, as it turns out, which is probably not highly desirable, but they all gave the same permeability value across different drugs to a first approximation. And then with that flow system that we were talking about, we came up with some mixing tank models and complete radial mixing models. And then we put those together to say, okay, well, given this flow and given these permeability values, what would we expect from this particular system? And then we did experiments, again, first on solutions, and then compared those two. They actually had very, very favorable comparisons. And then we wanted to, in terms of a system rate coefficient, we wanted to take a look at how that compares against in vivo. Since all these drugs that we're working with, we chose because we have pharmacokinetic data. So other experiments that we did were diffusion experiments of just the membrane, did some modeling of the system, did hollow fiber membrane permeation studies, compared that to in vivo also. So in terms of predictions, we measured some permeability values and came up with a mixing tank model. And then here were the predicted mass in receiver if we were to start off with 5 milligrams in this system. And the thing I want you to notice is, I mean, they're all more or less similar because, again, it's a system, for better or worse, doesn't discriminate between different drugs. But you can see here in 180 minutes, we're getting about 40% of the drug across. And that's quite remarkable. Again, this is the whole basis for going down this line. We were very motivated in coming up with an in vitro system where you can have complete absorption, ideally, because that does happen for many successful drugs. So that's kind of the basic challenge. And this was doing okay, I would say. Motivates us to do more, at least. And then so with this, with the model, in terms of this system, we wanted to look at, various issues, different things that we can control. If you envision back to what the system looked like, at least illustratively, we can have different flow rates. So here's the permeability as a function of flow rate. And as you might imagine, the permeability won't change. Permeability is a function of volume. Again, it doesn't change. Percent drug absorbed, permeating in the system after 180 minutes as a function of flow rate. It largely doesn't change, but it does go down a little bit at very low flow rates for the complete radial mixing model, which is expected. And most importantly, though, and this is blown up in panel D over here, the amount of drug that permeates in 180 minutes is very high for very low volumes. So at 900 mLs, a very typical volume used in dissolution, we're only getting about maybe even less than 10% absorbed, but we can make that go higher by having less volume, because essentially we're increasing the gradient basically by having lower volume. Okay. So those were all simulations to understand how the system would do. One major element was what are the flow rates that we use for in and out. If you remember, there's an inside part of each fiber and also the outside. And without going too much detail, we ended up using a flow rate of 2 mils a minute for each, the outside as well as the inside. This is the open circles here. So we start off with 5 milligrams. This was using metoprolol tartrate as a model compound. And then it goes down over 90 minutes to somewhere around a little more than 3. So we get, I don't know, maybe around 30% or so absorbed, 20%, more than 20% absorbed over 90 minutes. And then looking at the receiver, the open circle here, we see we get maybe 20 minutes, 20%, over 20% absorbed over 90 minutes using that flow rate. So the hollow fiber system is somewhat porous, so we can certainly have hydrostatic pressure effects. So for example, if we have a very high in rate, we actually lose donor volume, and we can actually just force the drug out of the donor into the receiver, and that was too excessive. So we ended up settling on 2 mils a minute for both donor and receiver flow rates. And then just comparing predicted and observed here. So across all drugs, which again, they have the same, very similar permeability. And there was good agreement between what the predicted was, whether it was the mixing tank model or a complete radial mixing model. The two are more similar than they are different. And we see the predictions and the observed are all very similar across these different drugs in terms of percent of permeating over 180 minutes. Again, we're somewhere around 40% or so. That's with 100 mLs, but we get a lot less than that, less than 10%, if we increase the, shall we say, the dissolution volume to the conventional 900 mLs. And then just comparing the flat sheet permeability values to the HFM membrane permeability values. They're made of the same material, but they're very different configurations. And we get the same values, including whether we have 100 mLs or 90 mLs of donor fluid. So our feeling is if we have a good understanding of the system with the accurate predictions, sufficiently accurate predictions, and just to compare this, how much we're getting across from the dissolution hollow fiber system versus alternatives, including some older ones. So here's a side-by-side diffusion system. It's very, very low because it has such a low area to volume. It has a low area in particular, including a low area to volume ratio. And then a long time ago, we did dissolution KCO2s, and it's extremely low, very, very low area to volume ratio, such that we get very, very little across. Has some utility, but not in terms of things that we want to do today. So here's kind of a major outcome of the first set of experiments. We're very happy with developing a model that was predictive and having good understanding, but then taking that and comparing this in vitro permeation rate constant, again, which is a function of the area to volume, as well as the permeability value. And this varies depending upon the volume, but taking the larger numbers here for a lower volume of 100 mLs, and they're at least on the same, generally tend to be on the same order of magnitude as what can be calculated from a human clinical study of a tablet or capsule. So here we have a value of around 0.6 per hour. This is 0.3 per hour. Lamotrigine was a bit higher. So this is off by an order of magnitude. Actually, and there's lots of reasons for this. Renine is a little bit lower, actually. Proxicam is definitely a bit higher. But in general, I'd say at least we're kind of in the ballpark. Having said that, there's room for improvement, including greater discrimination among different drugs with differing lipophilicity. And then meanwhile, other systems where there's a very low area to volume ratio, just very, very low, such that we probably won't say much more than that. Okay, so that was the first of two experiments, scopes, experiments of differing scope. So in the first one, I'd say we characterized things and we saw some degree of concordance between in vitro permeation constants and clinical permeation constants for various drugs. And the next thing we wanted to do is actually subject this, now that we have an understanding of the system and think it has some relevance, we actually wanted to do some tablet and capsule studies in order to predict in vivo human absorption profile and biopharmaceutic performance of, and we did this for 5 drug products using this system. So again, here's the same system that we're doing, that way we just described. All those prior studies involved solutions. Here we're actually dosing solid oral dosage form tablets and capsules. Okay, again, we have this hollow fiber system. We have a filter here, so there's a bit more going on than just what's illustrated here. But in general, we pump the dissolution fluid through the system. And in all cases, I think here we use 900 mLs, tried to be conventional in that regard. And then we have a receiver volume on the other side of the membrane, and then we can quantify what's in the receiver chamber. And the scope and flow of capsule studies, what we did here is, first thing we did is dissolution testing using just dissolution, that is USP2 testing. And then separately, we had previously determined the rate coefficients, in vitro rate coefficients. So together, we had all the information to at least predict how these tablets and capsules would then perform in the dissolution hollow fiber membrane system. So we did those predictions, and then did the experiments. We have observations about tablets and capsules in this dissolution hollow fiber system. And then we compared them, and there was very good agreement, actually. And then we had for all these tablets and capsules, we had human in vivo data, including their absorption profiles. So we wanted to compare those to see whether this in vitro system was capturing what was going on in vivo to any degree of accuracy. And then we also, in addition to looking at sort of absorption profiles through deconvolution, we did another type of analysis denoted here to be Levy-Poly plots. And we also, because they kind of inform a little bit about some rate limiting steps and biopharmaceutic risk and that sort of thing. And then we wanted to compare that type of analysis in vitro to the in vivo data. So just in summary, what did we observe? The predicted and observed absorption profiles in the in vitro system showed close agreement for all the different products that we looked at. The Levy-Poly plots from the in vitro system successfully predicted that 4 immediate release products, so of the 5 products, 4 were immediate release, 1 was extended release, and these to be low biopharmaceutic risk. Meanwhile, the plot from the in vitro system was successfully predicted that the metoprolol extended release product, and incidentally, one of these immediate release products was metoprolol. So metoprolol was both an immediate release product as well as an extended release product. But it was the extended release product that was observed to be and predicted to be higher risk. Okay, and of course the apparatus doesn't know what's immediate release, what's extended release. So in general, we would say at this point that this system has some predictive ability in terms of being insightful in terms of absorption kinetics and biopharmaceutic risk. And these are probably some more simple formulations compared to other types of more complex type of formulations. So it's what we have started to do now. So just to give an example, and this is metoprolol immediate release tablet. So here is a tablet just from USP, open circle. It's kind of covered up here, but it goes up very quickly and then plateaus out at around 100%. For the solution, we have a solution in the donor phase. Apply that to the hollow fiber system. All of it's dissolved, and actually we see a loss of it from the donor. This is 900 mLs because some of the drug is, of course, going into the receiver compartment, so it's leaving the donor compartment. And then we do tablet dissolution in this dissolution hollow fiber system. It goes up, and then it actually, just like for apparatus 2, but then it comes back down, very similar to the solution in the hollow fiber system. Okay. And if we would have done this in less volume, I think we would probably receive it. We'd probably go down much quicker. Again, lower volumes are going to have greater gradients, so we'll have more absorbed. This is the predicted and observed absorption profiles in the receiver chamber from the system for metoprolol IR products, IR tablets. This is the amount in the receiver chamber. So this is a solution, the closed circles here. So we have a solution, none is in the receiver chamber, and then it goes up over time, over the course of a couple hours. That's the solution. Prediction for the tablet is the dotted profile here, based off of the permeability of the drug and understanding of the flow of the hollow fiber membrane. And then the tablet, observed tablet, is the open circles. So the tablet kind of just trails a little bit the solution, but not too much of a trailing. So then what we wanted to do is we wanted to do some drug absorption analysis. And in particular, we wanted to take a deconvolution-based approach. And here's a model from quite some time ago to look at drug absorption. So this is drug absorption, fraction dose absorbed as a function of fraction dose dissolved. We see it's being modulated by the absolute fraction absorbed, as well as this term alpha here, kind of like a biopharmaceutic risk assessment measure. And if we just take this equation and we plot it, and we plot fraction absorbed as a function of fraction dissolved, here's what we get as a function of these so-called alpha values here. So if alpha is very large, where alpha is this ratio of permeation to dissolution. So if alpha is very large, it's sort of dissolution rate limited, and we get this sort of USP level A type of profile, where as drug starts off here, and then as drug is dissolving, it's being, relatively speaking, immediately absorbed. The opposite, and so that's kind of like a line of unity. opposing that is when alpha is very small here, this light blue, and we get relatively rapid dissolution, and then subsequent permeation, subsequent absorption. So this kind of looks like a reverse L-shaped profile. So we get this range of fraction absorbed, fraction dissolved profiles as a function of to what extent this alpha term is, where again, this alpha term is, as we'll see, is this ratio of permeation to dissolution. That is, to what extent is it dissolution rate limited? And so this kind of gets in this area of in vitro and vivo correlation. Just reminding you, the word correlation doesn't mean there's a straight line. You can have correlations that are based on linear, nonlinear functions. So correlation is just degree of relationship between two random variables, sort of how tight a relationship is, not necessarily how straight it is. And so I think some of you are probably familiar with Gerhard Levy, famous pharmacokineticist, came up with the Levy plot. In fact, this is from his 1965 article, Development of In Vitro Dissolution Tests, which correlate quantitatively with dissolution rate limited drug absorption in man, J. Form Sign 1965. And he has this fraction absorb versus fraction dissolve, but it's modulated a little bit. You can see it has T minus the lag time divided by 5.35. And back in the day, I think, can't say I was around, but it was quite a feat to do linear regression. Of course, that's less of an issue nowadays with software and computers. But there, the emphasis was on getting straight line relationships between absorption and dissolution. And I'd say this article is a good read even today. And by the way, Dr. Levy hit a series of articles on studying aspirin and was a big proponent of dissolution testing to differentiate high quality and lesser quality aspirin. But in this analysis, I think the thing that's maybe just different is that we're not trying to get straight lines per se. We're doing dissolution with the understanding that we take that as a measure of in vivo dissolution, and then we're doing, I'd say, biopharmaceutic drug absorption type of interpretation, where a major feature that we're getting is sort of the plot itself, and then with some implications about this alpha value here, which again has some inference in terms of to what extent is it dissolution rate limited or permeation rate limited, which has implications for formulation risk. And by the way, where does this model come from? It comes from arguably an overly simplified model of dosage form performance, where we have a dosage form. We have first order dissolution resulting in drug in the gastrointestinal tract, and then first order permeation to give drug in plasma. And sort of the little slick element of this is that, certainly we're familiar with dissolution being time dependent and absorption to be time dependent. But in this deconvolution analysis, we want to look at dissolution and absorption relationship. So we do that directly where time is implicit rather than explicit. It's a brief description of that. So again, just other assumptions in this analysis. We have only dissolution permeation going on. There's no metabolism, so to speak, although you could have some. Everything is, and those two things are first order, both dissolution and permeation. And then we're doing dissolution in vitro with the intent to mimic in vivo dissolution. And then on top of that, we also have assumptions built into deconvolution analysis itself. Again, the thing we kind of get out of this is this alpha parameter, which is a ratio of our two rate constants that we just talked about, permeation versus dissolution. So then if alpha is very large, we have dissolution rate limited absorption. It's small, less than 1, it's permeation rate limited. And then if it's around 1, then arguably it's mixed dissolution, mixed rate limited. So then looking at, so 5 products were actually examined here, just looking at maybe 2 of them, the 2 metoprolol products. And here is the Levy profile for metoprolol IR, both in vitro as well as in vivo as well as in vitro. That is the D-HFM system. At time 0, we're here. And then very early on, we have very high rates of dissolution. And then we have progressive drug absorption. So just going back to our family of curves here, we're getting everything that's here. So we're not seeing this part of the curve because we're just not sampling the plasma fast enough. So we're getting sort of a very extreme reverse cell type of profile. So we're getting, very, very permeation rate limited absorption product, at least in terms of this, again, this overly simplified model for dosage form performance, but clearly not dissolution limited. Meanwhile, the extended release products, and again, the apparatus doesn't know what's controlled release, what's immediate release. Here's a Levy plot for fraction absorb versus fraction dissolve for in vivo, which are the open triangles, as well as in vitro, the filled circles, and we get a little bit more like this line of unity, which you may recall is kind of more like the USP level A type of profile, where to achieve that you need high dissolution rate limited absorption. And as it turns out, this was an extended release product. So it's a higher, from a formulation standpoint, it's going to be a higher risk type of product. So we'd say the apparatus was able to mimic these profiles, which arguably have some meaning in terms of assessing risk. Okay, so that's a bit of a summary of these two papers. We're in the first one in 2022. We characterized this apparatus, developing quantitative understanding of it, and then applied it to some more simple formulations, although the modified release is maybe not so simple. But our goal now is to apply it to increasingly more complex products like amorphous solid dispersions, where drugs often have a very low solubility, and there's clearly we think, a dependence on permeation to allow for subsequent dissolution. Again, going back to the nature of the problem we're trying to look at. Okay, and then here are some additional references, and glad to take any questions. Thanks for listening. Okay, I'm taking a look at chat. Haven't seen any questions in chat, but glad to take any questions that you may have. Okay, so one question. I was wondering if you would expect permeation profiles in the system to be different if drug solubilization in bile acids like fed state bioavailable media was considered. There's been many articles that kind of relate to that. Not so much in this system, but other, I would say, very similar systems. And I think probably the biggest conclusion that's drawn is that the free drug concentration is what's contributing to drug absorption. That's not always the observation. So this is actually the first experiment that we've done recently. We actually observed that was not the case, that it was a system drug concentration that was driving drug absorption. So I want to say the literature is kind of a little bit non-conclusive, and our observation, at least with this system, was more in the minority. So I can't say I don't know of a unified theory on this, but is something I think that's a good question. We're equally interested. What are the linear dimensions of your apparatus, length and diameter? It's not very big. I kind of forget. I think the dimensions of at least the fibers are given in the paper, and the paper is freely available. But it's I'd say maybe about a foot long, so it's not very large. Again, I'd say it only has an area of 115 centimeters squared. So if someone wants to make a bigger system with more area, that would be helpful. It doesn't have to be a whole lot bigger, maybe a factor of 2 or 5 or something like that. That would be nice. What is the applicability of the system to other BCS classes? I think it depends a little bit on what you want to do. I would say the products that we studied were usually simple, including some being, for example, metoprolol, immediate release tablets are, I think I want to say they're relatively low risk products, right? The drug has high solubility, high permeability. Back in the 1990s, when I first arrived here at Maryland, there was a lot of expert formulators that tried to make bad metoprolol tablets, and they couldn't, at least within the realm of not putting them in concrete. So being a reasonable formulator, but not the world's best formulator on purpose, it was still hard to make bad tablets. But I would say it was, as was shown here, it was able to discriminate between different types of metoprolol products. And I do think some of the learnings have portability, actually, in terms of we, do analysis a certain way. But, as far as more complex products, I think it's going to get complicated very quickly. You may have noticed I had a hard time answering that question earlier about micelles. What's the effect of micelles? And I was expecting one thing in some recent studies that we did, and I was wrong in my predictions. So I think for us, we just need to do some experiments. Let's see, Matt Burke. Oh, I knew you would think of this, Matt. Can you put multiple D-HFM in series? And is a performance difference than when you concurrent flows the two liquids? I don't know. We haven't tried that, Matt. That's a good idea. Maybe putting a couple in series. That way we have a larger area to volume ratio. That's a good idea. Haven't tried it. Okay, Anna's a student here, and thanks, Anna. She put in some of the dimensions into the chat. Did we try a different membrane? How do you select the membrane material? Comparisons with the biphasic. So we haven't really done much of that. I'd say the only work that we've done was quite some time ago with CACO2, which has a limitation of area, I would say. Although I think since 20 years ago, I've seen people use larger surface area CACO2 for other purposes, but really haven't done much of anything. I think one of the slides I showed that Annette from Denmark put together, Southern Denmark University, there's really quite a few in the last couple of years. I don't know anyone that's actually tried all of those. We certainly have not. An advantage I can see of a lipid is because the one we were using is sort of a porous plastic, for lack of a better term, and all the drugs had a really practically the same permeability, which at this point is not a problem because I think area to volume is a bigger issue still, at least for us. But hopefully there'd be a day where we would start worrying more about the nature of the membrane. But don't have a really good answer. I think a lot of these things are new in the last, 3-4 years. I think there's quite a bit going on, and it's a bit diffuse. When we had an MCERC workshop in May, we talked about that a little bit, but it would take quite a bit of effort for someone to actually dig into all that. Let's see. Have you explored a fiber system where you can use lipid impregnated membranes? We haven't. That sounds like a good idea. Since you mentioned PAMPA, I just want to point out that Paul So, he was actually a graduate student here at Maryland. He got his PhD on PAMPA, where I think it's probably available. I think he published this. We published this, where we looked at different PAMPA compositions. So that'd be a good idea to come up with a lipid system. And I know from his work from some time ago, there's at least a suggestion about what lipids combinations to use. Yeah, Vasily's asking a question. Could you grow cells? Yep. In fact, people actually do grow cells with these hollow fiber systems in general for like making antibodies and that sort of thing. I would certainly worry about the integrity in terms of a monolayer. So if your question, Vasily, is, can you grow a monolayer? I think that would be, the integrity of the monolayer is what I would worry about. Um, but I think theoretically, yep, it's a good idea. Okay. See, we're coming up on at the University of Maryland. We start at the top of the hour and we end our lectures at 10 minutes of. So I think we're coming up to that time. Be glad to answer questions as long as there's questions, though. Do you think transfer dissolution coupled with your technique will be of impact? I think so. One advantage of the system I was showing you is that, the USP system is that's what we use. So I think it's possible for someone to use a transfer system or what have you. And again, an advantage of one of the reasons I have to say, well, we use this system over some of the lipid-based systems is because robustness to different media, potentially. So I think so, Suresh. But we haven't done any of that, though. Okay. One last question, maybe, if there's going to be a question. Hey, Dano, can you just put in the chat the MCERC email address in case people want copies of this notes?
Sure.
Okay, appreciate your time. Enjoyed talking to you about some experiments that we've done recently, and certainly enjoyed the questions too. I'm just going to wait until Dana is able to type in the chat the email address in case you want slides. Do you know what the email off the top of your head is, Dana? I actually forget.
Yeah, I'm added. I just wanted to make sure I copied it right.
Okay.
There it is.
Cersei at umd.edu.
Yeah, because
Yep,
off EU. Hang on.
.edu. Okay.
There we go.
Cersei at umd.edu. Okay, thank you very much. For those of you on the East Coast, have a good evening. For those of you in Europe, sleep tight. And those of you in Eastern Asia, have a good Wednesday. Thank you very much.